The overall goal of this procedure is to examine cupric oxide nanoparticle treatment as a contaminant removal process in production bleed water and to evaluate its cytotoxicity using cultured human cells. This is accomplished by first treating production bleed water with cupric oxide nanoparticles. The concentration changes of individual elements are evaluated before and after treatment. Next, concentrated cell culture media is diluted with either untreated production bleed water or cupric oxide nanoparticle treated production bleed water, in addition to standard cell culture supplements to prepare the test media. Untreated production bleed water media and cupric oxide nanoparticle treated production bleed water media is then applied to human kidney and liver cells in culture, and the viability is measured over 7 days. Ultimately, the MTT assay demonstrates the removal of arsenic, selenium, vanadium, and uranium from production bleed water by cupric oxide nanoparticles is associated with increased viability of human kidney and liver cells.
This is the first study to investigate the removal of specific contaminants using cupric oxide nanoparticles while assessing changes in cytotoxicity associated with the removal in a cell culture format. The main advantage of this technique over existing methods is that the environmental sample as it exists in nature is tested alongside the nanoparticles in an inexpensive, high-throughput format.
Cupric oxide nanoparticles tested in this study can be applied to other groundwater systems, such as domestic groundwater associated with small communities. The regeneration of cupric oxide nanoparticles creates less waste and minimizes waste disposal problems.
To begin this procedure,